Thanks for joining, everybody. Super excited to have Keri Kalmbach here to give a guest lecture on personalized medicine, and she already has her intro ready, so I'll just pass it off to her. Thanks, Keri.
Hey guys, can you hear me okay? Thumbs up, we all good? Okay, cool, and you're awake. Very impressive after a 3-hour sprint, so appreciate you guys coming. Give me a second so everyone can see my screen. Awesome. Okay, so this is a little bit different. So Erin, if you wouldn't mind, it's a little difficult for me to look at the code and look at you given all the faces in the gallery mode. So if you would just unmute yourself and speak over me, if there's like a question, if there are a bunch of people that are just like, oh, slow down, I would love to hear it. I'm used to having a little bit more of a direct line with the students, but feel free to speak over me.
Unmuting and speaking over people is one of my superpowers.
Oh, perfect. Okay, so who the heck is this chick? So I'm Keri. I'm only Dr. Kalmbach to people who really annoy me at work. But I finished my PhD at NYU School of Medicine back in 2015, where I studied biomarkers of reproductive aging. What does that mean? That means I worked at a fertility center and I took human eggs, I smashed them into tiny, tiny pieces and looked at the genetics and other biomarkers or just markers associated with these different cells and tried to predict fertility from women's outcomes. So I worked more generally, you could say I worked as a computational biologist. There got to a point where my spreadsheets were crashing constantly, and so I had to teach myself Python. That was right around 2012. And so that began a love affair with building and solving things in code and brought me to where I am today. Professionally, after I graduated, I worked at a small startup in New York City where we worked in genetics, and we developed what's called a gene panel, and we'll talk more about that in a minute, to better assess risk for diseases associated with infertility. I also led some of the largest studies in the women's health field and genetics. That was in collaboration with 23andMe. Do not trust their data. We can talk more about that later, but not good. Self-report, it's not great. But anyway, lessons you learn. In 2017, I moved to San Francisco. I was an instructor of data science and data analytics for a little known program called Mission U. It was a very ambitious little startup. So ambitious, they were acquired by WeWork in 2018. So I was then left to sort of find a different position. So I started consulting. Consulting just means that there are people who have problems but don't have a data scientist on staff. And so they come to my firm and they say, hey, I need a part-time data scientist to help me do this project. And so I work part-time on many, many different projects. I've learned so much. Consulting is a really excellent way to learn quickly and build up your resume right out of school or right out of your first job. So if anyone wants to hear more about that, you're welcome to email me or send me, you can have an informational interview. We're also hiring. So those of you guys who have for data analyst and data engineering roles. So I'm not trying to recruit any existing students clearly, but if there are any new students who are looking for, or recently graduated students who are looking for positions, feel free to hit me up. Okay, so why am I here? I freaking love teaching. My Mission U job was actually close to my dream job, to be quite frank. I enjoy the flexibility of working freelance as a consultant. I can kind of set my own hours to an extent, pick which clients I want to work with. And I've learned a lot, but I'm driven deeply by teaching. So you can probably see it in my face. Like, I'm exhausted right now, but I just light up when I get to do this stuff. So I really appreciate you guys being here, giving me the opportunity to teach something again, because it's been a while. Okay, so
I'm going to do the unmute and speak over thing. A few questions came up about 23andMe, which is it true that they keep data once you submit a test kit, and what are alternatives to 23andMe?
So the answer is yes, they will keep it. However, theoretically, there's a process of going back and requesting that they destroy your sample, but they have to consent to you. And so anytime a healthcare company, and yes, 23andMe is technically a healthcare company, they are required to go through a process of what's called informed consent. And it's actually a very, very involved process. And so you have to opt in to them keeping your data and giving them permission to give it to other people. So yes, they're going to keep it for their own internal purposes, but unless you give them explicit permission, they're not going to share it with anyone. And from firsthand experience, I can tell you they don't give it personally identified, well, as of 2 years ago when I was working with them, they did not give away what's called personally identifiable information. So that term has legal implications I don't need to get into, but they will give aggregate data. So they'll give the average age of a group of people. Will they give my age as a research participant to someone else? No, they can't legally do that. And then the other question was, what are alternatives? The genetics is good. So I'll say that. So the gene data is good. The problem is that when they make assumptions, think about we're going to talk about this later, but when you have, what are they assigning? It's all self-report. So let's say you have 200 people in the country who said they had bipolar disorder. I've got bipolar disorder. No, I don't. No doctor has, seriously, I don't actually have bipolar disorder, despite what you may think at the end of this talk. But in terms of the data, I could say, oh, my grandmother did. My mom was freaking crazy. Like, I bet I have bipolar disorder, even if I've never been diagnosed. And so the issue with their data is the fact that what they're calling their diagnoses are self-report, and they're not clinically confirmed. And so it's not the genetics. So if you want to know if you're a 50% match with someone who is your genetic relative, you can trust that because that's pretty good. But if they say you have a 50% chance of being born bald or a 90% chance of sneezing when you eat chocolate, first of all, it's not really useful information, but more importantly, it's based on self-report. So just keep that in mind. Does that answer your questions?
I think so. There are more questions coming in. Do you want to maybe get to more 23andMe questions after you dig into the data set a bit, or do you want them now?
Sure. So they'll actually learn a little bit about genetics through the process of this. So if it's about privacy, that's a little bit out of scope. But if it's about genetics, let's go through my mini biology lesson, and then we'll see where we get from there.
Sounds good.
Okay, so let's go ahead and get started. So let's talk about genes. I have genes, you have genes, we all have genes. And they're all stored on 46 chromosomes. I've got 46 chromosomes, you've got 46 chromosomes. But, I'm looking at Erin. Erin has darker hair. I've got blonde hair. How is it that we all have these chromosomes, but we're clearly very different individuals, right? And what does it mean when they say we've got 98% in common with a random monkey species, right? What does that mean? Think of your DNA as instructions. Okay, you guys are in a coding program, so you're familiar with the concept of like written instructions, right? So your DNA are written instructions, but instead of using our alphabet and the keywords that we use for like Python, say, there are only four terms, and there's A, C, T, and G. Okay, so you have 4 billion different base pairs in your genome. And every single cell in your body has roughly an identical copy of that DNA, right? Of the 4 billion base pairs. Now that's not entirely true. That's not entirely accurate. And we'll talk about that in a minute. So my question is, how is it that Erin has brown, I don't know what color eyes you have. It's like brown hair versus the blonde hair. Well, even if we have very similar genes, and he and I would be 99% similar, that 1% difference is in encoding those differences. But more importantly, every single cell in my body has roughly the same instructions. How does a liver cell know to become different than, let's say, a taste bud? Like how does that all work? Well, even though we have the same instructions, we have different genes that are turned on. Okay, so the only jargon that I want you to take away from this, I'm assuming you guys know what genes are, you understand the concept of DNA, that should have been review. I want you to take away from this the concept of gene expression, okay? So a gene expression is the combination of genes that are turned on. So a gene is said to be expressed if it's turned on. And so as you're developing, a liver cell will have a certain set of programs that are turned on. It'll have a different gene expression profile. So imagine just different pieces that are turned on versus, another cell like a skin cell in my body, right? And so what you can do is you can compare those two you can compare which genes are turned off, which are turned on and off, because you can have one that was on, but is then sort of in the process of getting turned off. And it's sort of like, how much is it turned on? Because you can also have it turned way up. Imagine like you can have a gene that has lots and lots of copies turned on versus something that is just not on at all. Okay, so I want to stop there. Are there any questions about that concept of gene expression? Because it is pretty critical to the study we're going to work on.
Doesn't look like any questions specific to that in the Slack, but feel free to ask them.
Yep. Or you can bribe Aaron and maybe.
One question. Are we talking about epigenetics?
No, we're not. Very good question. So for context, guys, epigenetics is a layer on top of the genes. And so that's a layer of complication that is a little outside of this, which is supposed to be kind of a bio 101, but, you know, points to whoever threw in the epigenetics. And I will say, you can apply very, very similar methods to studying problems with epigenetics like you can with basic genetics. So.
All right. And where are the on/off switches?
Great question. So depending on how you define a gene has different pieces, right? And so you've got what are called regulatory regions, and they're actually at the front of the gene. Then you've got the coding regions, which are in the middle, and then you've got parts at the end. And so it's actually pretty cool because a protein, something will come in and just bind to that beginning, and it's called transcription. And what it'll do is it'll basically say, it'll bind and it'll say, okay, you're on, there's an on signal here, which is a little beyond the scope of like how that happens. That's an epigenetics thing. Basically it's here and it's like, okay, I'm going to start copying all this. And so depending on those epigenetic signals, which, you know, I have a PhD in genetics. I could go into a lot of detail, or PhD in biology, so I could go into a lot of detail. My goal is not to put you to sleep after your long sprint, so I'm going to try to keep it high level. But if you are curious about it, you can Google differential gene expression or gene expression, that process, and it'll tell you a lot more about what's turned off and what's turned on. It's a very, very, very cool field. That's moving very quickly. Okay, any other questions?
Nope, we're good for now.
Cool. Okay, so I spent a lot of time working on genetics. Let's be clear, I haven't worked in genetics in 2 years, but it's way more interesting than the marketing stuff I'm working on now. So I thought you guys would find this more interesting. So I can also talk about marketing, but hopefully this is a little bit of a better choice. Okay, so let's talk about the problem we're going to solve in the demo today. I basically have an open source data set that you guys are welcome to play with. It's from data.world. I'm sure plenty of you guys have already gone down and like found my my read CSV statement down there and opened it up in data.world. You're welcome to play with it. But before we go into it, I want to give you a little bit of context about it. So basically, it's a fairly small data set compared to what, you know, some of the stuff you guys have been working with, but it's actually pretty reasonably sized for biotech. The reason being, every single row represents a woman who had a breast removed. Okay, so just, I mean, I know there are a lot of guys in this, but think about it. Like either that participant had breast cancer, and so she had her breast removed for reasons that were related to her treatment because it's a biopsy, so they wanted to get a sample of her breast, or she had a total mastectomy because it was part of her treatment. Or these are women who had either breast reduction surgery. So that was our experimental group, is the group of women with breast cancer. Our control group are healthy women who don't necessarily have, they didn't have cancer, but maybe they thought they had cancer, or they had breast reduction surgery. Okay, and so the data that we're looking at is basically we have a baseline, okay, and we've got healthy tissue, and then we've got cancer tissue on top of it, right? And what we did was we said, imagine that there's a matrix, and we have signals, and we know that certain genes are turned up and certain genes are turned down, right? And what we did was we said, okay, what's the difference between the healthy and the unhealthy, or the cancerous, right? And what's that difference? How different are those gene expression profiles, okay? You can think of it as like how much is that gene turned up in cancer or how much is it turned down relative to the baseline. Okay, so the data set itself is filled with women who are cancer individuals, but I needed to sort of like, one way to describe it is normalization. If you guys are familiar with that concept, where you normalize a value so that it's on a different scale. Similar thing. So that's the gene data we're working with. Okay, the data itself includes only cancerous individuals. So questions we can answer with this include, with this type of data, are like, is the person, if we have cancer, is that person likely to survive? And that's actually the question we're going to answer in part today as part of this project. 2, we can answer questions like, is treatment X likely to be effective? Right? Because genes actually as you guys know, play a large part in whether or not a particular radiotherapy or chemotherapy will actually work, will actually act on that type of cancer. Can also help with just diagnosis. You can do a descriptive analysis. You can do clustering, if you guys are familiar with that concept, and you can describe different types of cancers based on those gene expression profiles. So there's obviously tons of money being thrown at this, both in the private and public sector. I'm only touching a very, very small part of it, so that you guys can just get a taste of some of the things that you can do if, you know, with your new data science chops, you want to go, I don't know, save lives. I mean, it's not ambitious or anything, but you can do some very, very cool stuff with the stuff you're learning, or will learn if you're a junior person. Okay, so the question we're going to answer today as part of this demo is, does the addition of expression information from a single gene improve the accuracy of cancer survival prediction? Okay, so rephrasing this, we've got baseline data. Okay, we've got any information a cancer patient would have in her EMR data, in her EMR record, her electronic medical health record data. So for instance, we'd have her age, because obviously maybe not obviously to you guys, but if an individual who's 45 has cancer versus someone who's 85, maybe the 85-year-old isn't going to have as much a chance of surviving because they're older, right? We also have things like the diameter of the tumor that they found. Larger tumor, bad, right? So we're not going to go into every single column. I invite you to look at the data yourself. But basically, we've got this baseline data. This is the information that's available to the physician, okay? And this is what the physician would use to make decisions about treatment. The question we're trying to answer is, on top of that, if you add on a genetic marker, can you more accurately predict cancer? In other words, we have a baseline prediction, and we're going to calculate the accuracy of our model. When we add genetics to it, or a single genetic marker, does that improve the accuracy or not? Okay, so this is something that I spent a lot of time working on. But instead of, when I was working with the 23andMe data, it was dozens to hundreds of genes. For sake of a simpler example, we're only talking about one gene today. Okay, I want to stop there. Do we have any questions on that so far?
Yes, some questions have come in. So does the 0 or 1 value in amputation refer to which breast?
That is a very good question, and I genuinely don't remember off the top of my head. I apologize. I can, however, go back to the original paper and look that up.
Sure. And as you said, we can dig into the specifics of the features perhaps outside the scope of the presentation, but there's lots of data there.
Yes. Well, I'm going to acknowledge by saying I don't know the answer to that, but my instinct is to say yes, because some women may just have had a biopsy, which is basically just like a pinprick pulling of data, so they didn't necessarily lose their whole breast as a consequence of their cancer.
All right. And then most of the other discussion has to do with, it was provoked by a question: how do you predict survival in treatment when treatment options improve every 3 to 6 months?
Ooh, well, I guess I need some clarification on that question. Are you talking about the fact that there are multiple treatments of an individual patient, or are you talking about more generally the fact that treatments are changing quickly as a field?
I think it's more generally, and the argument that's happening in the channel is that supposedly the data is not stationary, whereas some people are saying that maybe you could still get some valuable insights even if the treatment itself has changed.
But
Because
the way
after all, what you're looking at is the genetics.
exactly. So what we're looking at, so the genetics you can treat as we in the biomedical field, we would make an assumption that it's relatively constant. However, it's not. And so what we're doing is that's why you want a larger sample size, which is obviously important. Treatment is a tricky moving target. The other complicating thing about trying to predict effectiveness of treatment is that oftentimes doctors don't participate in studies. And so it's what's called observational data, which means that you are biased because you, instead of randomly assigning a treatment to someone in these types of genetic studies, oftentimes to get the largest, what you're going to do is you're going to say, okay, who are those people that have already had this treatment? And then study their background. And so what I first described was a randomized controlled trial, which is very, very hard to do in genetics for a variety of reasons, because you can't randomly assume people are going to have different genes. So you can't really assign them properly to groups. So it's all observational. And so you have that bias. So very good point. It is a very, very difficult thing to study.
Sure. But to clarify, for what the question we're asking right now is, it's focusing specifically on the genetics and less on the treatment. Is that correct?
Well, yes, exactly. So for our particular question, we're this is just one snapshot. This is one study, and you would never change clinical practice based on one study. And we're talking about survival rather than the treatment itself.
All right. And then the last question I'll go ahead and ask from this is, why not survival analysis?
Actually, that's a very good question. So survival analysis is one of my favorite analyses to do. So survival analysis is about, so if you were predicting someone's survival, that's literally what survival analysis is. Because, and just to sort of paraphrase for some people or explain what survival analysis does, is it says it's a model where it takes at any given time the fraction of people that have either survived or died. And so you'll have these really kind of morbid to say, but cool curves. It's kind of more morbid because you're talking about survived people dying. But you can really see the trend over time of people that are dying versus those, and the rate at which they're dying, depending on different treatments. More practically, you can use survival analysis for churn analysis for clients. So that's, yeah. So you can use survival analysis, but the question we're specifically asking is whether or not we can predict we can improve the accuracy of our of whether or not they're going to survive.
Gotcha. And I guess I'll throw in one more question, and then we'll get back to things. Does model type dictate sensitivity so much that you would not see it if you picked the wrong model type?
See what?
I see if you've picked the wrong model type, I assume. Vincent, if you want to clarify. The effect of the additional feature.
Yeah, that's a very fair question. So if this were an actual research study as opposed to, an hour-long discussion, we would definitely do a lot more than what I did here. This is just a snapshot. But what I will say is this, when I worked, this is very similar to the approach that we took. The reason being that random forest as a model type, which for those of you guys who don't know, I'll explain in a minute, is actually really flexible. It is fairly efficient in terms of computational power, and it actually was it gave us the most accurate results. So I picked random forest because that was what we used at my last job. And so although I can't say I was the scientist who was responsible for trying those early iterations of the model, what I do know is that at the end of a year of modifying this data and working with it, trying neural networks, trying all, regression, all kinds of different approaches, the random forest was actually the best approach. So.
All right. Well, thank you for entertaining the many questions, and I'll let you get back to it.
Yeah. Well, I mean, to me, the code is less interesting than the conversation, but you guys are here for code, so we'll talk about it. So that said, there are a lot of other, before I dive in, I was saying this to Aaron, there's a wide variety of different people that are here, from expert Android developers that I met earlier to just, you know, just students who are just starting to people who are fresh graduates. So I tried to keep it fairly basic so that everyone would be able to have the same vocabulary. I think the interesting thing is less about the approach I take, because random forest, I mean, at least half of you have done that before, I'm sure. I want you guys to appreciate the application of it and in solving a problem in the field. So I'm not going to apologize for the simplicity of it, but just explain to say that trying to manage a diverse audience. Okay, so first thing we're doing, importing pandas. Yay! So for those of you guys who are not quite as familiar with it, pandas is the main data manipulation library. Didn't exist when I was teaching myself Python in 2012. It had just gotten started. I had 2,000 lines of code. Pandas is awesome. I can now recapitulate my 2,000 lines of code in about 100 in data cleaning. So just saying, love your pandas. What we're doing here is we're creating a new data frame. All I'm doing is linking directly to the data.world page. They allow you to do that. I'm reading the CSV and I'm dumping that data into a data frame called DF. Okay. I'm going to go through just a couple of housekeeping things. The first thing you want to do when you look at a data set is how big it is. What are you looking at? Make sure it's the correct shape as to what you're expecting. Another thing you should do is look at the first couple lines. So df.shape tells you the dimensions. So we have 272 rows and 1,500 columns. Seems like kind of a weird balance, doesn't it? We'll get to that in a second. The other thing I like to do is look at the head. And actually, this is a very clean data set, but it would also be worth looking at the tail. If you're pulling from a CSV file, oftentimes there can be like blank rows. And so if you do df.tail, it'll sort of tell you if there's any trailing white space. I know this is a clean data set, so I'm not going to worry about that. Okay, so we have a lot of information here. We've got patient number, we've got a kind of an arbitrary ID number, we've got age, event death, survival, time recurrence, chemo, hormonal, amputation, histotype, diameter, the grade of the cancer, which is level of severity, generally, I think it's usually between 0 and 4, lymphocyte infiltration, angio infiltration. So this tells us whether or not there was blood, whether angiogenesis had happened. And so in other words, the cancer was so far advanced that it had actually started pulling its own blood supply. Lymphocyte infiltration means how much was the immune system responding. And then ESR1 is our first gene. So I want to orient you guys to everything after ESR1 is genetic data. So we have the name of the gene sometimes. Other times it's just, I mean, literally contig 45645 in biological terms means random snippet. And so not every gene has a meaning, not every gene has a function, or every piece of information here is associated with a clean genetic identity. We don't necessarily know what that gene does. And there's a lot of it. So what do we have, 1,500? For sake of this, so for sake of this demonstration, I am actually going to limit the number of features, okay? So I actually wanted to put this out to the students. So we've got 272 patients, and we've got 1,500 features effectively, right? We're not necessarily gonna use all of them, but why would we wanna limit the number of features? Just as a kind of a philosophical question.
All right. One of our instructors said, if you can't answer this, I will be upset. So, all right. Stephen Sinclair says, the curse of dimensionality.
Very good answer. A couple of other considerations.
And several people are typing, so we'll add a few other thoughts. Vincent says it's below the 5 times n or 5 times n squared problem.
Very cool. That's a statistical rule of thumb. That's a good one.
And let's get a few more. While people are coming in, somebody did ask a question as well. What are the values for the gene snippets, normalized on, off in that region?
Yeah, so on would be positive if it's getting turned, if it's turned, So think of it this way. It's on a continuum, right? You're going to have a high level of expression or a lower level of expression, and the baseline is the normal tissue. So if there's less of that gene turned on relative to the baseline, it's going to be a negative value. If it's a positive value, it means there's more in that cancer cell than there is in the normal healthy cell. On average. This isn't, because we're...
All right, and then let's two other responses to your question came in: overfitting and interpretability.
Yes, I love those. Thank you guys. Okay, so all of those reasons. Personally, and this is a matter, and DS instructors, feel free to push back on me. I like to curate my models, and I mean, all of those are very valid reasons. But how would I limit these? I want to pick the ones that speak to my hypotheses. So as a subject matter expert, someone with a PhD in biology, I'm going to make a little bit of a assumption here and throw out some of the things. Like, I'm going to assume barcode has nothing to do with survival rate, because barcode is literally the ID for the specimen in the biobank. So it's just an index for the sample. Things like ID, probably just an arbitrary identifier, right? So I'm going to throw that out. You don't have to be a subject matter expert for that. But I like to start with smaller models and then build them out, which is why, for sake of this example, we're actually going to start with just one gene and see how that influences our model. Okay, cool. So the first thing I'm going to do is I'm going to do a baseline model. And so here, I don't really know how much detail to go into, but effectively what I'm doing here is I'm only keeping the data that's in the first 17 columns, right? Then I do the describe method, which will tell me, it gives me what are called descriptive statistics. You guys know you better know what descriptive statistics are, otherwise I'm gonna be very upset with your instructors. Mean, median, and mode are the most common examples, right? And that will give you, at a glance, an idea of what's going on with some of these characteristics. Like, what is the average value? Like, what's the average age of this data set? The average age is 44, which is really interesting, because the average age of a woman who gets diagnosed with cancer is actually like closer to 65. If you look like at the broader cancer population in the United States. So that's something that's worth noting, because maybe this is a biased data set for reasons that we don't really understand. So it's important for context, but it's also important to eyeball it to make sure there aren't any outliers, right? This event death, where if we're assuming 1 means there's a death and 0 means they didn't have a death event, 28% of our individuals died. Yep, and okay. So we're going to limit the features. I'm excluding a couple of these. It was a little bit of a judgment call. If you guys are interested later, I'm happy to add some of these back and play with the model a little bit more, but I want to keep it simple. So all I'm doing here is I'm saying, okay, I only want to include a certain set of features, and I'm going to assign that to a value capital X. Okay, and that's a data frame, a multidimensional data frame that only includes the features. Y is our response variable. It is what we're predicting, right? So that's going to have our event data, so the death data or not. Now, when I run this, and of course, okay, and actually, I want to ask another question. What's going on here? Why am I breaking this up between a test and a trained set? Even you newbies should know this one.
People typing.
Sure. I know. Sorry, guys. I know you just spent 3 hours in a test. I'm trying to try to keep your brain working.
All right, model validation. We can create validation sets to see how much our model is overfitting.
Yep. Yep. Yeah. Yes.
To check how well the model fits the data and to validate.
Yep. To validate is a good answer.
There's one word that I think people would be good to include.
Which is?
I'm going to fish for it too, maybe. We'll see. All right. I'll drop it, but people who are typing can keep typing. Generalizability, I think that's a good way to think about these things.
Yes. So to paraphrase, Aaron, correct me if you have a different opinion about this. Generalizability means that if we set this model out in the wild and try to make actual predictions, we want to make sure that when we're evaluating it for validity, in the test and train sets, that it's applicable, that it's randomly assigned. And so we are randomly assigning different values into these different testing and training sets. And the reason that it needs to be random is so that they are systematically the same. The same reason that if you do a randomized control trial in an experiment, you want them to be exactly the same, except for whatever you're trying to, whatever variables you're looking at in the experiment. Okay, so just to be super explicit, so we divided it up. I kind of arbitrarily picked a test size of 0.2. If you guys want me to do 0.3, happy to modify that. It's a little bit about convention. Actually, in biology, they pick 0.5 for some reason. The features we're looking at are age, whether or not they've had a history of chemotherapy, hormonal treatment, amputation, and I will look that one up. Some other diagnostic markers like a histotype, which for our purposes doesn't really matter, the size, whether or not they're positive nodes. So positive nodes are, if you have cancer, you've got lymph nodes, which are kind of like, think of it as like they're sentinels for your immune system. These nodes are where if there's an immune cell that responds to your cancer cell, it will actually it'll take that message back to the lymph node. And that's where the immune system actually sort of organizes its response. And so positive nodes means that it's generally bad. And only and most of them are going to have positive nodes. Grade, angio, I already explained these, but okay, cool. So let's get into the more interesting stuff, guys. I know we're already 45 minutes in, but thank you. And this is where hopefully I'm not going to spend as much time because you guys should know this a bit more than the biology. Okay, so I am importing a classifier. It's an ensemble method. And I'm going to throw one more question out there because for sake of time, I'm going to limit it. But what's an ensemble method? Why would I choose this versus something else?
And while people respond to this, some questions came in about stratifying or making sure the training set has similar proportions of values to the whole set.
That's an interesting question. Because we're only talking about 272 samples, I would not want to further stratify because you're only going to make the sample size smaller. However, a good practice in biomedicine that I didn't do here is to look at the demographics of your different sets. So like what I really should have done to make sure that they're similar is I should have, for instance, compared the average age and make sure that they were both right around 44, right? Because otherwise, maybe there was someone who was 105 and who was biasing one of the sets, right? And so very good call, and I did not do that in this particular example. And so to be clear, stratification is sort of like when you try to match groups together, and it is something that's done when you have really large sample sizes, but stratifying inherently whittles it down. And so because you only have 272 or 273 samples, I wouldn't want to do that here.
And also, I think I'd reiterate what you said earlier, that this is a somewhat simplified demo for the scope of this lecture, and that makes perfect sense. But these are great, clever questions, and I'm glad that our students are thinking so critically, because these are the questions you'd have to ask and dig into to do these things even more deeply. So some of the answers that came in for ensemble is that an ensemble combines predictions of several base estimators with the same learning algorithm. It's supposed to improve generalizability. That's close, I think, if you want to give your take on what ensemble is.
Well, I would actually I wonder if is there a more, that's a very technical answer.
Somebody said a weighted average of many statistical learning methods.
But why? Why do we want to use ensemble? That's what I'm trying to, oh dear, my runtime disconnected. I'm happy to fill in, but.
To improve generalizability is one suggestion, and to prevent overfitting.
I think that what I was looking for was you're combining multiple models. And so in doing so, you sort of broadly, and I'm not here to teach you about ensemble methods. I'm sure that's what unit 2 or section 2. It helps you because you have more information. You're having one model and you're taking that information and improving on it. And so by combining multiple models, you get a better model and a more accurate model.
And some answers like that are actually coming in now too. So yep, to improve predictive performance.
Yes, thank you. That's what I was looking for. So why, yeah, I guess maybe I should have emphasized why ensemble as opposed to what is ensemble. I did ask what is ensemble. So thank you for answering what is ensemble. Yes, you guys should get your cookies or whatever it is that you give these guys for giving correct answers. They're stars.
Usually emoji.
Emoji. Okay, cool. I'm gonna give you 2 literal thumbs up. Thank you for those were very technical, very good answers. Okay, cool. Matt, so let's go ahead and dive in. We've got 10 minutes effectively because I do want to be able to have a conversation about this. These are data visualization libraries. You guys will learn more about that later. Broadly, what I'm doing here is something called hyperparameter tuning. And so for those of you guys who are Android developers, for instance, what is hyperparameter tuning? They're levers to pull to make the model fit better, right? And so an out-of-the-box model will fit and can make predictions, but you can improve on that. And so what I've done here is I picked something called the depth range, or the depth, I should say. So a random forest classifier is an ensemble method, which means it's a bit of an aggregate of something called a decision tree. And what the decision tree does is it basically makes splits in the data. And it says, okay, if I split on this feature, let's say we split at age 44, right? Just picking that number out. And we say we're going to make a call based on that decision as to whether or not someone has cancer or not, right? And so it keeps going down. So, okay, we have age as one informative feature. Let's say angiogenesis infiltration is the next. And it kind of randomly goes through and tries to evaluate these cuts and whether or not that divides up. And as you get down to the bottom of the tree, it tries to find the best tree that will assign an identity to or make a better classification for what we're looking at. So in this case, we're only trying to predict death or not. And so as you can imagine, if we have a tree that is too deep, and here's a quick question, because I know you guys will get this. You've said the answer about 6 times. Why would we want to, wouldn't it be great if we could just go all the way down and learn all the details of our data? Why is it a bad thing if there's one word I'm looking for, and I know all of you guys know it. What is it?
Overfitting with 4 exclamation points.
Yes, sorry. I'm not trying to be obnoxious. Okay.
I think it's just enthusiasm.
Yeah, okay, good. Okay, so overfitting is real. And so in the wild, you do need to do your hyperparameter tuning. I'm gonna let you guys read this. This is more about the application and less about the actual code. You have the code. Feel free to email me. I'm happy to walk through if you have any questions. Just using a for loop, which you guys should all know, and iterating through a series of different values for the depth of the tree and trying to figure out what the best fit is. Okay, and so what I've done here is I've calculated my test score and my training score. And ignore that. And now I want you guys to interpret this for me. How would we optimize, like, if these are our values, our testing and training score, what max tree depth would you guys recommend? And why?
All right, so people are saying 9, 8, somewhere in there, and see if anybody's giving the Y a stab. 10, so 8 to 10. Somebody says it's a good example of early stopping. Test score is clearly decreasing after 10. 8 is the highest testing score before variance gets too high. The minimum highest score. Look for local maximum.
Yep. That's okay. You guys have got it. So just glancing at this really low resolution image, you can't really tell the difference. 8 to 10 looks good. The training score always goes up for reasons you guys probably understand, because it's got all that information. So it's just going to keep learning and improving on the training data. The testing score is the better assessment of its performance in the wild, right? Because the model is naive to testing data. And so both are important. But what I look for is sort of like, because sometimes the training score can actually go down. Not usually, but sometimes I've seen it. And so I kind of look for the minimum distance. But so you want to maximize the testing score, as you said. But I also kind of like to minimize the distance between the two. Sometimes it's a little clearer than others, especially if there's like a plateau point with the training score, which we're getting pretty close to 1. So that's going to be a plateau. Okay, great. So I would completely accept 8, 9, or 10. And as you can see, I sort of arbitrarily picked 10. And said, okay, that's the value we're going to use for that particular that hyperparameter. And so I'm going to plug that in and say, okay, what is our baseline model? So remember when I was like cleaning this data up, all I did was pull the baseline EMR data. So so this is age, chemo, things the physician has in the electronic medical record, okay? And I ran that, and I got at a max depth of 10, we got a training score of 0.97 and a testing score of 0.745. So now, with 10 minutes left, I'm going to give you just the quick answer, or the quick next step. The question we were trying to answer was, how does addition of one gene or one genetic signal improve the accuracy of our model? So what I did was, this is the original code. And the way I did it was I was basically taking the original set of data, and I dropped ESR1 before, but now I have it. So I included it. I inherited it into this feature data frame. And so when we run the exact same model again, we get a testing score of 0.65, and we get a testing score of 0.75 as the baseline. How do we interpret that? And this is where I would love to have more of a conversation.
All right, let's get some people typing.
And that's the end of the thing. So I'm welcome. I invite more conversation.
Let's discuss this, and then it's also open to whatever general questions you'd like to ask.
And actually, by the way, this happens all the time, guys. Like, this is real data.
So somebody said you are overfitting by adding the gene.
Okay, potentially.
Gene confounded the existing model.
Could you define confound?
Passing that back to Vincent.
Well, no, just because in biomedicine, confounding has a very specific meaning, and I just want to be sure that we're saying that.
I'm assuming Vincent means it in a more general statistical sense, but I'll let Vincent speak to that. Okay. Maybe Vincent is a biostats person, I don't know. Mm-hmm. So somebody else is saying, first, we can programmatically add each gene and combinations of genes to see if there's a sweet spot of genes that improve accuracy.
Yep. Yes, indeed. And that would 100% be the next step. I think what we would probably all agree on is that ESR1, based on these results, it's not conclusive as to whether or not ESR1 actually improves our diagnostic accuracy.
And Vincent defined confounding, saying it covaries with some of the variables in a way that reduces predictability.
Yes, so that may be true. So another way to think about that, and correct me if I'm wrong, Vincent, in going the next step in that thought, is if you add more data, you're kind of adding noise. You're not contributing a useful feature here. And so that actually decreases the testing score.
Yep, and Vincent agrees.
Yep.
Nico has a question about this. Would performing something like PCA on all the genes be useful, or is it necessary to keep the gene information separated?
That's very, very clever. So actually in those 23andMe studies, the first step was always PCA, because there's a lot of exogenous variability that you can't really overtly control for, especially since you can't really randomize people. And so PCA, when you do these models, truthfully, you always have principal component 1, principal component 2, and usually up to about 5, depending on the size of the data set. But yeah, PCA has a large, large role to play in these genetic studies. So very good call.
All right. Floor is open for any questions or any other comments on next steps and last stages here. All right, questions coming in. What is the method to optimize which genes and how many will improve this score? That's kind of what you were discussing, but.
Well, I mean, you could use a kind of classical feature engineering approach. Personally, I would consult a subject matter expert first to sort of at least prioritize the different. So I would take an academic approach, which is to go look it up and try to figure out which genes are likely, inform specific hypotheses. But that's based in my approach as an academic, someone who is studying this formally. If you were working for a company, they might want something more efficient. And so you can actually just use your standard approaches for feature engineering to evaluate which features are important and those aren't, by sort of putting them in and taking them out and looking at whatever error metric you want to choose.
All right. I'm sorry.
No, I was going to say, but the one thing I wanted to say is that if you start doing an additive model, like just adding one in and taking one out, adding one in, taking one out, is pretty standard. Once you start getting interaction terms, like you start saying this one interacting with this one, it gets computationally prohibitive, because it becomes when you talk about 1500 data set, 1500, and this is just one, this data set's from 2010. This isn't even like one of the largest they have. Like there are thousands of these. You can't study it that way because we just don't have the computational power to match the dimensionality of that data.
Yep. And to our students who are at the very end of our curriculum, there's some CS computer science material. Like it's a little bit into Big O, which I feel is at least philosophically related to what Keri just mentioned, and it's good to think
Absolutely.
about. So some other questions. Do you consider yourself more of a data scientist or a bioinformatician? And what do you think the difference is between the two?
Oh, I can tell you exactly. So I'm a data scientist now. And like I said at the beginning, this is actually drawing on previous work experience that I had. I haven't done biology work in about 2 years, to be completely candid. So I consider myself, my job title is senior data scientist. I just figured you guys would find this more entertaining than a marketing problem. Although if you want to have me back again, I'm happy to talk about marketing and actually survival analysis. That would be a great little thing to talk about. So someone who is a bioinformaticist or bioinformatician, can't remember. Those are people who study the genes themselves. And so they will actually do like the alignment. So for instance, that normalization step, that was developed by a bioinformatics person. So where they actually took the raw genetic data. What I'm doing is more what I would call clinical data science, which is where I'm just taking the higher level data. I'm taking the clinical data, which is like the age, the clinical parameters. I'm not actually getting into the genes themselves. I am a little bit, so it's kind of a mixture of the two, but it would definitely be more leaning towards genetics if I were a bioinformatics person.
All right. If you use PCA, do you have to make sure you have as inputs to the model the original inputs? So you have to test potentially unneeded genes.
Well, so I'm probably not going to answer the question directly. Conventional use of PCA in this type of study is actually to okay, now that I think about it, I understand what you're talking about with PCA. So you can use PCA to sort of like summarize the different dimensions of the genes, right? And try to find different patterns that way, right? When I was talking about PCA, the way they actually use it, and so what that will actually tell you is something a little bit more interesting. It'll tell you the groupings of genes that are associated with different things. And that is incredibly informative. And personally, I would say that one thing to take away from this is that a single gene doesn't contribute a lot. And so when you start doing that what's called pathway analysis, of which PCA is one approach, you start getting way more powerful. It's kind of like if you're studying something on the zip code level, it may not be that useful, but if you start going up to like aggregating to a statewide level, the path level, you can start to see how different features there's a lot more information that's converging at that state level that will give you a more powerful prediction. So, so to answer the question, any single gene probably isn't likely to have a lot of impact, but in biology, people just tend to throw all of it in there and see what comes out the other side. They're not that careful about which genes because you're talking about thousands usually.
All right, and then one last quick PCA question is, would TSNE be useful along with PCA? Which I believe is a visualization.
I actually,
approach.
maybe it's just because I've been out of it so long. Could you define that abbreviation? I'm sure I've heard of it. I just don't.
T-distributed stochastic neighbor embedding, and I'm digging into it myself now.
Oh, good. I don't feel quite so bad. So, off the top of my head, I can't answer that question.
That's fair. And to people who ask about the answer, the standard data science answer to would such and such an algorithm or approach be good is, I don't know, test it. At least
Yeah.
that's my standard answer. Like it may well be some clever people. Jeffrey Hinton made this one, so it's probably clever.
Well, he probably has
But
a grad degree in biology. So,
yeah.
yeah.
All right, some other questions. If we are overfitting on very wide data, would it be worth doing ridge regression?
If we were doing a regression,
Ridge regression
yes.
for this wide data.
Yeah. I mean, that's one approach to, um, yeah, that would be applicable.
I
Personally,
guess in general, what are some other machine learning models you would consider when
oh,
evaluating the question at hand?
oh, okay. That makes more sense.
Well,
So,
I'm combining questions at
okay,
this
got
point.
it, got it, got it. So there's a lot of interest in neural networks right now, understandably. One of the interesting things about sort of what I would call classical machine learning approaches, things like linear regression, decision trees, even things like SVM, sort of like the classic, the things that you saw in Andrew Ng's intro to, data science from 10 years ago. You can't throw more data at those. So it gets to a point where they're of diminishing returns where, like, if you throw more and more data at it, it actually plateaus. However, neural networks in recent years have clearly demonstrated that you can actually throw way more data, and there isn't a point, it doesn't really have the same sort of diminishing effect. So there's a lot of interest in neural networks right now. Frankly, physicians and scientists are very, very conservative, and they're really suspicious of the hype that comes from tech. So they're not going to be using, except for a couple of labs, candidly, are not going to be using the sexiest, newest things in data science, which I think is only to the benefit of you guys, because you come in with that knowledge of the sexy new stuff, and you can apply it to these other problems. So that actually gives you a competitive advantage to someone who has a PhD in biology, for instance.
All right, I'm going to ask just one more question, which is, you mentioned how wonderful it is to have pandas here in 2019. What did you use before pandas for data cleaning in Python, and do you think any library will replace pandas anytime soon, or other libraries you like using for
A
this?
hell of a lot of for loops was what I was using before pandas. Unfortunately, I it's kind of, well, I don't know, maybe it's a generational thing, but I don't see myself leaving Facebook anytime soon. Like, there's a certain amount of like commitment because you've got that investment in it, right? And pandas, for most of the stuff, just kind of works. I know, like, someone showed me a graphical user interface that's built on pandas recently. I don't think the concept of a data frame is going anywhere.
Oh
I don't
yeah.
the basic features. Is there going to be a better, quicker, more efficient, easier to learn syntax that comes out? Oh, absolutely. Not in the next year, though. Not that I'm aware of. Nothing I've seen so far. Well, R
Yeah.
replace Python? No. That's just
Well, yeah, but I would add exactly. I mean, and some people are also saying this data frames have been around, and pandas basically is data frames for Python. So there are a few interesting ones that seem to be trying to be more performant or whatever, and I could see more progress in those directions because pandas is a bit of a hog sometimes, but you can't beat it for workflow, at least in my opinion too. So, all right, thank you so much. I guess I lied. I'll ask one more quick question, which is email or best contact for any
great.
ongoing questions.
So I'm actually just going to put it in the chat here. I will add it to that link in the notebook too, so you have it there. But
I'm not sure if there is chat in the
there is
Zoom.
no chat. Okay, so it's just my name. It's keri.kalmbach@gmail.com. But I will be sure to put it in that first like intro slide or intro cell in the
Of the
notebook.
notebook. Sounds great. Yeah, people have the notebook.
And guys, even if you just sort of like want to network about and to be clear, they're not hiring for data science jobs. They're hiring for senior data analysts and data engineers. So just want to make sure we manage expectations. For my consulting thing, I'm happy to talk about biology. I'm happy to talk about my transition from biology to whatever. I'm happy to have a coffee over Zoom.
Great, thank you so much. And I would emphasize to our job searching students that both of those sorts of roles are a perfectly fine way to break into this industry. So I think that that's a great opportunity to check out. Thank you so much, and thank you everybody for sticking and asking so many questions and conversation during this presentation. I'll have the video up later this afternoon.
Thank you so much, guys. I really appreciate the great questions.
All right, thanks everybody. Bye.